Can we really define life? Do we really understand it? Does life match a rigid set of criteria? Or is there life out there that may go beyond our understanding?
What if we find some sort of exotic entity that stretches the boundaries of our definition of life? What if we find something which in some sense is living, but in another sense is non-living?
It'll be like discovering a lost continent or something like that. It'll be a whole new field of biology.
It's an entirely new universe of things at this very small scale that was completely undetected before. And who knows what other planets could have very small living things.
Perhaps we are made from bits and pieces of our solar system and cosmos. We could be from Mars.
Ancient rock found its way from Mars and it got to Earth. And today we are on the threshold of establishing that life might have existed at some point on Mars. We're now on a doorstep to the heavens. What a time to be alive.
August 1996. NASA made a sensational announcement. We had an alien in our midst. This nondescript-looking rock was a 4 billion-year-old Martian meteorite. Originally discovered in the Antarctic, it was to become the most famous chunk of rock in the cosmos. Inside, there were strange worm-like formations. NASA claimed they were the fossilized remains of ancient tiny creatures. This, they believed, was evidence that life may have existed on Mars, and perhaps still does. Evidence that we are not alone.
We began to ask each other, is this for real? And I can honestly say that evening when I went home, I had difficulty sleeping because of the thoughts of what this could be, and it's undoubtedly the most exciting thing I've done in my 27 years as a scientist.
The international euphoria was tremendous and short-lived. According to the world experts, the so-called fossils could not be the remains of living organisms. The issue was their size. They were simply too small to live. Half a world away in Australia, a young geologist, Dr. Philippa Ewens, was about to uncover extraordinary new evidence that would put NASA's case for life on Mars firmly back on the agenda. Her discovery would set her on a collision course with the international scientific community. Philippa was working on a consulting project for a petroleum company, doing standard analysis of rock samples. She was using a powerful electron microscope, one of only a handful in the world capable of magnifying objects up to 1 million times. Little did she know, this routine job was about to reveal a hidden world right here on Earth. For she was about to stumble across something new and utterly strange.
I'd never seen anything that looked like this before, which I could account for as some sort of mineral. There was this whole range of structures, some of them sort of swollen blobs on stalks and stuff, and they were growing in the very small spaces between the clay. At first I thought I had a very strange rock, But as we went down a sort of series of steps to show what it was, I then realised that it wasn't mineral. It had to be some sort of organic material.
The size of these structures was extraordinary. Many measured less than 200 nanometers in diameter. That's 1/1,000,000th of a centimetre, or 1,000th the width of a human hair. Intrigued, Philippa went in search of the literature.
I went home and dug out all my textbooks on botany and biology and microbiology to see if I could see how big things were, just to sort of show, well, this isn't this because it's too small.
Philippa's strange structures were 10 times smaller than any known form of life.
I just had to know what it was because there was nothing described in any of the books about bacterial fungi with these dimensions. And then it was like, well, if it's this small, what is it?
And then, back in the lab, something very odd seemed to be happening.
I started to notice that the side of one of these Petri dishes was a fingerprint that was white, as if it had been like dusted with something. And I thought, well, that's a bit curious. Why am I getting this white stuff actually in the plastic, and what's this fingerprint?
Under the microscope, the same strange structures appeared to be growing from the rocks onto the Petri dishes. Could this mean they were alive? Surely they were too small. Then Philippa made the connection to the tiny formations within the discredited Martian meteorite. They were 10 times smaller than any known form of life. Ultra-small bacteria, nanobacteria.
How many here have ever heard of nanobacteria? Good, okay, this should be a lively debate. Now, if you want to have fun, we should...
Professor Kenneth Nielsen of NASA's Center for Life Detection is a world expert in tiny life forms. He is also one of Philippa's most formidable skeptics.
When someone goes a factor of 10 and confronts everything we know about the theory of living organisms, then we have to deal with that. My first impulse is to say that it's impossible. The second one is it's unlikely, but I'm always glad to be proven wrong. But it needs proof.
It all comes down to size. A cell with a 200 nanometer diameter is considered the minimum volume necessary to house the essential components of any living organism. A creature 10 times smaller simply cannot contain the essential components of life.
What I would try to make you remember is that the nanobacteria is smaller than that. So now you have to figure out how you take all of this structure and make it disappear down to something that's this small. There are several groups who claim to have living organisms in this size range. And what I would challenge you to think of is what that organism would have to be like if it was really there.
Philippa had found structures below the 200 nanometer threshold for life. Some were as tiny as 20 nanometers. She christened them nanobes. Like microbes, she argued, just smaller. But if they were indeed like microbes, that meant they were alive. If they were alive, then they had to be some weird new life form. Philippa believed the nanobes had all the trappings of living organisms. The task ahead was to prove it.
Following a rule of 200 nanometers is not something that's going to put me off trying to find out more about them. So it just makes them even more intriguing.
Philippa pulled together a crack team to study the nanobes.
The main thing.
Dr. Tom Loy, paleobiologist and world expert on ancient DNA. Anya Yago, geologist. And Rick Webb, microbiologist.
Cut them. It's brand new. Then great, you know, this is a really, really big thing to do. But I'm a skeptical sort of person, and I have to have all the evidence there before I'm willing to go with it.
first reaction was, oh, yeah. But the evidence that Philippa showed me with just the pictures was pretty convincing. And so I just made the assumption that, yep, these things are alive. Let's figure out how they're alive and what they are.
The first step was to find out what the nanobes were made of. Rick produced cross-sections that revealed what appeared to be a cell wall and an area containing nucleic acids. Further tests indicated the nanobes were composed of carbon, nitrogen and oxygen. And there were preliminary signs of the key marker of life, DNA.
If they're not alive, I have no idea what they are. I can't explain what they are. If they are a rock, and they have all these properties which are characteristic of living cells, then they're really strange rocks.
Strange creatures from a strange world, an alien underworld. 3 to 5 kilometres below the Western Australian seabed. Down there, the heat and the pressure is tremendous. Few living things can survive temperatures of up to 170 degrees Celsius. Nanobes would have to be very tough indeed.
The nanobes are surprising at every turn. The environment they came from is deep under the ocean floor. It's very hot, thousands of pounds per square inch of pressure. And they're old. They've been around. I mean, they're in geological formations now. Unless they've somehow worked their way all the way down from the Earth's surface that deep, I suspect that we're looking at a time slice in the past. Jurassic Park kind of stuff, but with bacteria instead of dinosaurs.
Philippa believed she was hot on the trail of a new life form. A big claim, especially for a geologist, a rank outsider to the biological club. It was time to put her data and her reputation on the line. Time to go public. Her paper was published in the journal American Mineralogist.
Breakthrough study finds new life form.
Philippa and her nanobes became instant media celebrities.
Form of life, microlife, has just been announced at the University of Queensland. Dr. Philippa Ewins says they are so minuscule that theoretically they shouldn't exist.
They're turning up in the most extraordinary places. These so-called nanobes might be
The similarities with the tiny Martian nanobacteria rekindled international debate.
The origin of life and the possibility of life on Mars or elsewhere in the universe continue to make headline news around the world. Here in the studio to discuss some recent developments are three people who've been following the subject closely. Malcolm Walter, Philippa Ewins and Michael Payne. Well now Philippa, you've been studying rocks not from Mars but from Earth for some years, and you've found features that superficially at least look very much like those in this Martian meteorite. Now what about the problem that these little objects are just too small to have all the necessary bits and pieces necessary for them to be living?
Well, that's exactly right. That is the big question. How do these small cells function? People now are beginning to think, well, maybe they have some entirely different biochemistry which is allowing them to operate, to reproduce somehow differently. So
these are like alien life forms that you've discovered.
Well, I'm not sure they're alien, or just something very different, but it's...
Professor Paul Davies, physicist and astrobiologist, led a chorus of skeptics.
Any claim of that sort has to be greeted with a certain amount of skepticism. That's right and proper in science. We're dealing here with an extraordinary claim, and to use the dictum of Carl Sagan, the late Carl Sagan, extraordinary claims demand extraordinary evidence.
The real debate here is there functional life at scales that most of us can't imagine? And if it's there, what the devil is it? You know, and it's something new.
In a way, I hope she's right, because if she is right, it'll be profoundly significant. We'll discover a whole new form of biology that at present we can't conceive of. But I don't think that's going to happen.
Speculations, ideas are cheap. hey, what if the universe is alive? What if the universe is expanding 100 times a second and collapsing? You know, in science, if it's not testable, it's not science. And so if you have a weird, wacko idea, and I think research is all about it. Research is we don't know what we're doing. That's why it's called research. But we're playing around with ideas, but we also have the requirement we have to have hard, reproducible results, and we have to be in control of the variables.
You risk being, I guess, being thought of as a bit strange. But, you know, I guess you could, you know, if you got it wrong, you could risk your career, but I don't ever worry about things like that. I just sort of do follow my nose.
But you haven't got it wrong.
And, well, exactly, I haven't got it wrong.
Across the Pacific, a maverick professor put his career on the line over a decade ago. Emeritus Professor Robert Falk was the first to suggest that nanobacteria actually exist.
When I first started coming up with these little things about 1990, my colleagues all thought I was nuts. In fact, one of my dear colleagues considered this as a career-busting fiasco. And the real boost for it was when NASA found these nanobacteria in meteorites. And then people began to say, well, there may be something here. Maybe I'm not so crazy after all.
Like Philippa, Professor Falk has discovered nanoorganisms in rocks. But he goes further, arguing that there is an entire realm of nanobacteria existing everywhere. He believes they are possibly the dominant form of life on Earth.
The previous conception is that big bacteria are the world's greatest biomass, but I think these guys are perhaps 10 times even more abundant than big bacteria. So if this were true, it would make the bulk of the world's biomass consist of nanobacteria. And I think this is a kind of an important discovery. I think this is a very exciting area to be working in because it's an entirely new universe of things at this very small scale. It was completely undetected before. Just look at this picture here. I mean, any microbiologist will recognize this long thing as a well-known iron bacteria. But look at all this stuff in the background. How can they deny that these are some form of living organism? We've got a dozen different sorts of things here. It's a veritable zoo. And why don't people realize that what's going on here anyway? Is this life or not life?
Structure does not prove life. That's what I really think of this work. We're not used to looking at this size scale at living organisms. And so we get down to this size scale and we see something that looks like a bacteria, which really is pretty nondescript, isn't it? It looks like a little small cigar. Lots of crystals look like that. Other things look like circles. By golly, they can be crystals too. So the onus is on the person who has seen the structure to tell you what it's made of. Then one might believe that it's alive.
I've never gotten any funding for this nanobacterial investigation. I mean, if I were running a big lab, yes, I could do all this stuff about finding their metabolism, what their waste products were, this sort of thing. But I'm too old to set up a lab to do this. This requires a big lab project, lots of assistants, lots of money, and so on. I simply don't have the facility to do that. I'm saying, look here, biologists, here are these things. You guys have got the facilities. You take it and run with the football.
Back in Australia, Philippa was running with the ball.
Hi, Anya. Hello. How are we going? Good.
In the laboratory, the team was witnessing some remarkable developments. The nanobes appeared to be reproducing and growing larger.
Since we've managed to get them into liquid cultures, we've got a much better idea of how fast they grow. We get not a lot happening for about 1 to 10 days, but from 12 to about 25 you get active growth and an awful lot of activity. You can see with an optical microscope all these cells going absolutely crazy.
This video is from a weekend of working on one slide, just looking at it and watching it and trying to figure out what we're seeing. And it's really weird. Like, you've got this outer sac here that's wobbling around, and then there's an inner one with membranes, it looks like. Who knows what it has to do with this thing next to it. I've looked at all kinds of microscopic things, strange stuff, and this is totally unlike anything I've ever seen before.
They're forming these crazy morphologies, but we have no idea what it really means. I mean, what the heck is this? You know, I mean, it looks like a tire tube or something bizarre like this.
And these ones are really good. What we have is a nanobe soup, a mix of small ones under 200 nanometers and also a mix of larger ones. And it may be that the small ones are associated with the large ones or maybe become the large ones, but we don't know that yet, and that's what we're working on. Totally bizarre.
To convince the skeptics that the nanobes were alive, they had to establish the existence of the most important ingredient of all, DNA. Rick was hunting for the telltale black dots that would signify its presence.
It was about 5:30 on a Friday afternoon that I finally got a result. And I was looking in the microscope and suddenly I saw them. And I was ecstatic. I was jumping around the lab. There was nobody else here. I was by myself. It was incredibly exciting at the time. We could now say, there's nucleic acids in these organisms. You can't tell us they're not alive anymore.
The presence of DNA was significant, but not conclusive. There was always the threat of contamination. It happens all the time. A few common bacterial cells from the lab bench or water supply infect the sample and produce the wrong DNA result. Philippa's team would have to take the next critical step, extract the DNA and determine the genetic sequence. But that would take time and money. So far, the research effort was coming almost entirely out of Philippa's own pocket. Her need to be taken seriously took on a new urgency. Her best hope of attracting funding was to be published in one of the respected biological journals. She failed.
In one case, Philippa got a rejection from one of the very prestigious journals saying, oh, this research is too controversial, we don't publish controversial research. You really, I mean, that in itself really makes you wonder, like, what is the point of any sort of scientific publication in journals like that? If the paper follows scientific method, it must be worth publishing, even if it goes against existing paradigms and existing ideas and all these sort of things.
With the amount of work and the costs and the time and everything, and this was always done when lots of other things were going on, it was just really, you know, what does it take? Yeah.
In the far north of Europe, Philippa discovered she had an ally who was also out in the cold. A Finnish scientist claims to have found his own breed of nanocreatures, not in rocks, but in the human body. He believes they are making people sick.
My personal opinion is that nanobacteria are dangerous to humans. However, we don't know yet how dangerous and who is under risk. It is obvious that nanobacteria are very slowly growing, and if they cause a disease, it may take many years to develop.
For the past decade, Dr. Olavi Kianda has been investigating the presence of nanobacteria in blood. He is convinced they play a critical role in triggering kidney stones.
So just now we have injected nanobacteria, and you can see...
Through his experiments with rabbits, he claims to have discovered that nanobacteria pass from the bloodstream to the kidneys, where they cause the formation of stones.
90% of the kidney stones we analyzed contain nanobacteria. We can see small particles around 100, 200 nanometers, sometimes smaller than that.
Dr. Kianda believes that nanobacteria form protective shells of calcium around themselves. Like pearls form around grains of sand, he argues, kidney stones form around nanobacteria. The medical significance could be staggering. Dr. Kianda believes nanobacteria may trigger other conditions that involve unexplained calcium deposits, such as Alzheimer's disease and arthritis, some cancers, and heart disease.
If we are right, then we might be able to solve 1, 2, maybe 3 or more diseases, how they are aggravated, triggered, or even what is their main cause.
Dr. Kianda's work has been published in some of the most prestigious journals, and he has even developed commercial test kits to detect the presence of nanobacteria. But, like all researchers into life at the nanoscale, his work has been met with skepticism and outright hostility by the scientific community. In 1999, he received the most lethal professional blow. He was accused of scientific fraud. Not because his nanostructures didn't exist, but because he claimed they were alive.
All such attacks make the life miserable because you have to defend yourself, you lose your grants, you lose your credibility. Your publications come back with a letter that this is ridiculous, go home. Well, it doesn't really make you feel good.
After 2 inquiries, Dr. Kianda was cleared of fraud. But the battle to convince his peers that his nanobacteria are alive continues. He is encouraged knowing Philippa is engaged in a similar struggle.
If you think early era of microbiology, it was much more easier because people were having their eyes open. New findings were present in every journal, every issue. Now new findings are rarely present. So to introduce a new finding puts an enormous task for providing evidence of all aspects of the organism. And that is sometimes ridiculous.
Well, it's nice to know that there are other people around the world that are finding similar things, and I think now let's just get on with it and just see if there really are these little bugs in rocks or in, in tissues or in people, and what are they doing?
In 2001, after 5 years of ongoing skepticism and mostly self-funded research, Philippa's data was demanding attention. She was awarded a large research grant to sequence the nanobes' DNA.
The fact that we got this funding supports our idea that these are living organisms, because we wouldn't have got that kind of support and the level of support we got to do DNA sequencing of a rock.
The funding raised the stakes and Philippa's profile. She now had access to a top microbiology lab where they would run the tests.
Normally, we just tend to do our PCR setup on the bench, but however, I just want to be really sort of cautious because we're working with this novel isolate, and just get ourselves one of those racks, pipettes. These are what we're going to be using. I'll probably also use...
A lot was hanging on the results, not just proof of life, but vindication of years of work. But what if the nanobes didn't respond to standard DNA techniques? What if there was contamination? Expecting months of tests, amazingly, they got a result in just over a week.
I really wasn't expecting a result, so we were all crowded over the table and had a look, and then Deirdre screamed, "Ah, it's worked, it's worked." And Anya jumped in the air and screamed too, and so it was just a really fantastic, exciting moment because it was so unexpected.
They were hands up in the air and hugs around, and it was the best feeling.
The fact that it worked just blew us away.
A pair of unassuming looking bands of DNA provided proof. There was life in the sample, and it appeared to be bacteria.
This is what we've got. We've got a result, and that's it there. And
When they looked further, the nanobes appeared to be not just one organism, but a community of organisms.
we didn't expect just one template to be there. I mean, I would have been a bit worried, like, you know, because then that probably would be contamination or something, you know, you just never know. Yeah. But there's definitely something there.
That's brilliant.
More than one.
4180 change.
It's a huge leap for us because now we have some information that we can use to try and silence some of our skeptics.
We've achieved in the last 2 weeks probably more than we have in the last couple of years in terms of the molecular side of things. But this is just a big breakthrough.
To the results. Exciting result. Fantastic.
A breakthrough? Maybe. Evidence of some sort of life? Yes. But exactly what sort of life? The next stage was to analyse the DNA sequence and find out exactly what it was.
If we're dealing here with some exotic side branch of life as we know it, then there ought to be some overlap between the genes in you and me, or cockroaches or geraniums or whatever, and the nano genes. On the other hand, it could be that we're dealing here with something perhaps a bit simpler than the type of life that we know and love. And that would be truly exciting if that was the case, because it would help unravel the whole problem about how life got started in the first place.
Nanobes challenge our fundamental understanding of how life began on Earth. Charles Darwin believed that life emerged from the primordial soup of elements, a freak accident of chemistry where non-life became life. Could nanobes be part of that transitional stage?
Imagine in investigating these structures as we get to smaller and smaller scales and we look at them, we find something that is not really alive, but is kind of alive. It has the features A, B, C of life, but has the features D, E, F of non-life. So in some sense, it might be a missing link or a series of missing links. And that's really the business that we're in, because we'd like to know how that transition occurred.
Seeing as the origin of life is one of the great outstanding puzzles of science, anything that shows us the pathway that led from non-life to life is going to be enormously significant.
But what if nanobes, or their relatives, played a role in a far more radical theory? That life came from space, ferried to Earth, inside rocks.
We know rocks come from Mars to Earth. There's a traffic of material from there to here. Today, I think it's increasingly accepted that it is possible for microbes to ride inside rocks from one planet to another, in particular from Mars to Earth, and survive the journey.
Professor Paul Davies has spent a decade exploring the likelihood of microbes surviving space travel.
The first danger is getting off the planet. In the case of microbes escaping from Mars, for example, they'd be subjected to accelerations of several 100,000 G, enough to kill you and me in pretty short order. But being so small, it turns out that bacteria, for example, can certainly survive these sorts of enormous accelerations.
Deep in outer space, microbes would have to withstand extremes of cold and radiation. But cocooned inside a rock, in a state of hibernation, they could feasibly survive for millions of years. Their final challenge would be surviving entry into the Earth's atmosphere.
I think that, that even conventional bacteria, albeit of a hardy nature, could make the journey from Mars to Earth or Earth to Mars without too much difficulty. Obviously, nanobes would have the advantage as spacefaring organisms, if that's what they are, because they're smaller, and so they could be subjected to more violence, more intense radiation, and so on, and survive it.
Dozens of rocks have successfully made the journey from Mars to Earth. NASA's Martian meteorite that had caused such a sensation back in 1996 was one of them. The alleged fossilized nanobacteria it contained supported the theory that microbes can and do travel through space. but only if you could first prove that life could exist as something so small. In the years since NASA's announcement, Dr. Everett Gibson, co-leader of the Mars meteorite team, has been building the case, challenging the world's skeptics to consider life outside their standard criteria.
Can we identify life? Can we recognize life? Can our narrow little box that we define life in, can it be expanded? Can something else be put into the mix, whether this be in a rock, whether this be in a fossil, whether it be in a meteorite from Mars? Because if we can, we've answered the big question. We are not alone in our solar system. There could be life somewhere else.
Dr. Gibson has collected every press clipping on the subject and remains a true believer in the face of scorn and derision.
I'm very proud to say we as a team feel stronger today about our original hypothesis than when we wrote it in 1996. Of course, we have our critics because that's what science is all about. The more radical the idea, the more attacks that come towards it. Whether we're right or wrong, we really don't know that yet because it's an ongoing detective story. But we feel that we are right. We're answering a lot of our critics, but we open up new exciting areas in science, and that's what science is all about. This is a win-win for science because we're going to be better prepared when we go to Mars. These are incredible.
Philippa's nanobes bring the debate back to Earth.
Clearly, if one can prove they contain DNA and what's the sequencing of them, it makes the story very interesting and complicates some of our concepts of what they really are, what life biology is.
Philippa has come a long way. The evidence is mounting that she may have stumbled across an entirely new life form. Nobel Prizes have been won for less. She has been invited to present her thesis at a prestigious international conference concerned with the detection of life on Earth and beyond. The audience will include the most distinguished names in the field of astrobiology. It's time to meet her harshest critics, face to face.
Some people think size doesn't matter, and Philippa has discovered these features in rocks from Western Australia and now elsewhere, which she calls nanobes. Rather non-committal word. They are about the same size as the features in the Martian meteorite, but in this case, we're not talking fossils. If Philippa is to be believed, we're talking about live organisms.
The gauntlet is thrown down by Professor Stephen Hyde of the Australian National University, who suggests Philippa's nanobes are nothing more than crystals or silica gardens, rocks that grow.
Now watch this. Looks very alive, but it ain't. They're also very similar to the morphologies at a different length scale to what Philippa Ewens is seeing. Here's one of her pictures. And the general morphology is very reminiscent of these silica garden type effects. Basically, it's bursting open a membrane, the salt is rushing out with the silicate, and so you're getting almost a chaotic growth. But there is a preferred direction, which is, well, I'm not saying that yours is a silica garden, but I'm saying if it were, you can see we always get this preferred, the filament, if you like, is just due to the jet.
Oops, sorry about that. If we go up in magnification here, we're at 22,000 times. We can see these unbranched filaments sticking up into the pore space. We get these large rounded structures as well, and we see these growing in between these plates, and this one here looks as if it's actually put enough force on here to break that particular plate apart. So at this point, we can say that nanobes are communicable and grow. They're membrane-bound structures with cell walls, cytoplasm, nuclear area. They're non-crystalline by another technique. And just a few weeks ago, we got very excited because we saw we had bands here on the gel, which meant we'd actually successfully extracted DNA from the cells and amplified it. So this is a great breakthrough for us. We were really excited about it. It sort of opens up which way to go next, because now we know we've got to try and isolate them out, and that's what's going on at the moment. Thank you.
she's come a long ways, and to see it to be presented before this astute scientific audience and see the criticism, but not all criticism. There's praise for the work that she's doing now, which is nice to see. I mean, it's just that she's turning around perhaps some of her disbelievers.
Yes. I think what they might say is, though, okay, we've extracted DNA from an environmental sample, but what about we need to show the little ones.
We want
pure cultures, yeah. So every step we get to, they say we want another step.
I'm still very sceptical, especially after hearing the talk by Stephen Hyde and seeing all those very biological-looking structures that he produces completely non-biologically.
Ah, Philippa, I'm looking for you over. Would you mind if we have a quick look through your lecture?
No, as long as you keep
Charlie Limeweaver.
Hi, Philippa. Hi.
You've not met.
Better grab some chairs.
Right, thanks.
In a back room at the conference, Philippa is asked to provide a private presentation.
That's what they look like. They are really bizarre.
I'm looking at a bunch of objects. What is...
What we have in the cultures is a complete mixed bag.
Why are there holes in the middle?
Because they're really strange.
Wait, these... If you look at this, I said, that's an nanobe and that's an nanobe. Is that what I'm supposed to...
None are here. You can't actually see any nanobes because they're in the background, but the nanobes are associated with these large organisms. It's like a community of...
Oh, so the name nanobacteria really is inappropriate for... I mean, you have a zoo of 1,000 different creatures of various different sizes, and we have this one word, nanobes, which we say, oh, it's something new, but in reality, we have a whole continuum of creatures which...
Well, the nanobes themselves may be part of a continuum of a larger group, or it may be that they are separate, yes. Yeah.
So, presumably, you're not the only person in the world doing this.
There are other groups also saying that they've seen them in these sorts of environments. I'm not sure who else is actually working on living material. Kayanda's working on it, yeah.
They claim to have found them in blood.
Presumably, there are some disease-causing agents in the nanobacteria world.
Finnish people believe.
Yeah.
And wouldn't that necessarily say, hey, you guys are finding something new?
Which amounts of money.
Yes. Why aren't you just flooded with money to try to identify what these things are?
Well, I am being.
Oh, great.
Recognition at last, from an unexpected but very welcome quarter. Several $1 million of investment is marching Philippa's way.
We're here today to meet with Ian Jones, who's flying over. We've been talking with him for about 1.5 years now, and he's a potential investor to take the nanobe work further forward in terms of commercialising some of the properties.
Finally,
yeah, been a long time.
An international biotechnology company has taken notice of an extraordinary development in the nanobe's research. The original Petri dish, with Philippa's distinct fingerprint, has undergone a dramatic transformation. The nanobes seem to have a bizarre appetite for plastic.
This was one of the original infected plates. We've taken a sample out of that to infect others, and you can see it's frosted on both sides. This is not just surface erosion, this is actually burrowing into the polystyrene itself. The amazing thing about it is that this is all happening with absolutely no nutrients other than the plastic. The surface of the plastics, as the degradation begins, there's just a light little streaking. It looks sort of like somebody's just kind of brushed it with a bit of sandpaper. But then as that develops, the parallel tracks go along for a ways, and then they turn around and they come back. Whatever is responsible for the track avoids another track. Either goes under it, or they go away in different directions. It's got to be a complicated arrangement how they're doing it. Oh, I thought this was really something, because if we've got something that can live on plastic, and especially polystyrene, here's something that takes the nanobes out of being scientifically and interesting to possibly commercially interesting, but as well having some benefit to the world, getting rid of all the polystyrene cups that are in dumps and all around the world.
Hi. Ian, nice to meet you.
A deal is being struck. It's not what the nanobes are, but what they do that is important here.
When you first saw them in the rocks, were they active like that?
No, they were just...
They were just dormant?
They're just sort of sitting on the rocks, so they're not crawling around all over the rocks like this. It's only since we got them into media and at high temperature, although I haven't actually observed the rocks at high temperature.
That would be interesting.
That would be interesting, wouldn't it?
Yeah.
See, these chaps are interesting. They've got the little ones inside.
Oh, yes, look at that. We're looking for all the sorts of things that a nano does. One of the things it does is eat plastic. We have to find out what else it eats. Does it eat petrol? Does it eat coal? Does it eat other minerals? Now that is sufficiently science fiction-like to want curious people like ourselves to want to investigate further and find out what are they doing which could be useful to mankind in developing industry, protecting the environment and doing other things. 20 years from now, we could have a whole new range of industrial or environmental processes. We might be able to, for example, have a nano spray which you can just spray on plastics, for instance, and would chew up all the plastics.
It's really good to see that the business side has seen something here. They know it's controversial, they've done work, they've looked into the science, and they're prepared to take the risk. So it's good to see, after all the scepticism.
Yes.
The investment company is planning to provide significant resources to fast-track the nanobes research. New laboratories, new equipment.
This is a significantly high-risk investment for the company, but like all early-stage scientific research, we could come up with the jackpot, hundreds of millions of dollars, or we could come up with nothing. If we are successful in developing the nanobes, Philippa should do well out of that financially.
To fight tooth and nail to get like $5,000. Yeah. Money doesn't drive me. I'm not expecting to get rich over these. I'm sort of more driven by wanting to know more about them. But on the other hand, if it's commercialised and millions of dollars are made and I just sort of struggled along and don't get anywhere, then equally I don't want to find that that's the situation.
Tomorrow we'll go through the schedule and particularly have a look at the research agreements and some of the simple management aspects and things like that, and start the ball rolling on the patent side.
As soon as we get the go-ahead, we'll just put people in and then just do everything. So that's going to be, then when that contract gets signed, it's going to be great.
Incredibly, the party is over before it's begun. The biotechnology contract has collapsed, victim of the international money market. And more bad news. The DNA sequence of the nanobes has thrown up more questions than answers. There's a tantalizing trace of something you'd find in a common microbe, but it implies contamination. The skeptics will have a field day. So, where does this leave Philippa? The bad news might be good news. If the sample has been contaminated, it does not discredit the work. It just means they must repeat the tests. And if it isn't contamination, the results may be more profound. Perhaps the reason they could not conclusively identify the nanobes is that they are a new, as yet unidentifiable life form. That's what Philippa believes.
If it is a contaminant, who knows?
Oh, if it's a contaminant, then it's the world's most interesting contaminant.
Yes, absolutely.
Curiosity has now become obsession. There is no turning back.
If you are a scientist studying elephants and you're looking only for elephants, that's what you find. It is extremely important for scientists to keep their eyes open so they can see new things.
If nanobes are real and they are living organisms, then I think it could be tremendously significant. In fact, if it turns out that these really are some sort of missing link between the non-living and living realms, this could be one of the most significant discoveries in the last 100 years.
Philip is taking the sort of risk that a lot of innovative scientists take. If she turns out to be right, she'll be remembered as a pioneer. If she turns out to be wrong, she'll be remembered as a crackpot.
Can we define life? Is there life at dimensions that defy our understanding? Whatever they are, nanobes and their relatives are taking us into uncharted realms. They may be the thread that unravels the mystery of the origins of life. We may find that just as space is boundless and infinite, perhaps there is no limit to the ever-diminishing smallness of being.